Influenza epidemics affect millions of people every winter. The human influenza virus is an enveloped ellipsoid-shaped virus of the Automyxoviridae family. This virus attacks respiratory system cells. The viral genome consists of 8 RNA segments enclosed under the viral membrane. Each segment is associated with nucleocapsid proteins and the polymerase complex. Hemagglutinin is an envelope protein located on the surface of this virus. It plays an essential role in viral entry. It is organized in trimeric spike, each spike comprising 2 subunits called HA1 and HA2, connected by a disulfide bridge. HA1 has a globular domain which interacts with the cellular receptor. HA2 is protected by HA1. It has a short sequence of hydrophobic residues called the fusion peptide at its amino terminal end. It is anchored in the viral membrane via its carboxy terminal end. This subunit exhibits long helices oriented perpendicular to the membrane. This helical architecture is characteristic of class I membrane fusion proteins. Viruses are simple biological compounds that need to infect cells to replicate. Influenza virus first interacts via its hemagglutinins with salicylic acids anchored to the surface of the respiratory epithelial cells. This interaction initiates virus entry into the targeted cell by endocytosis. A vesicle coated with clathrin is formed and released into the cytoplasm. Transport vesicles are driven by molecular motors inside the cell, where they fuse with early endosomes. Proton pumps present at the endosome membrane acidify the vesicle lumen. Below pH 6, this acidification results in important conformational changes in the hemagglutinin structure. HA1 moves away from HA2. Loop B, connecting helices A and C, rigidifies and becomes helicoidal. As a consequence, the fusion peptide is projected away from the viral membrane and anchors in the endosome membrane. Helices C and D dissociate, and helix D leans onto helix C. This new rearrangement pulls the viral membrane towards that of the endosome, and the 2 membranes fuse together. A fusion pore is created, and the viral genome is released into the cell cytoplasm near the nucleus. The released viral ribonucleoprotein complexes carry nuclear localization signals. They enter the nucleus, where the viral multiplication process begins. New viral particles are synthesized with the help of the cellular machinery. They bud off the cell and infect new cells. Imifenovir is an antiviral molecule that blocks the fusion mechanism. It carries an indole core and can bind strongly to hemagglutinin, impeding its flexibility. During the acidification process of the endosome, hemagglutinin with bound imifenovir can no longer change its conformation. The fusion mechanism is blocked, and the influenza virus remains trapped in the endosome. The resulting late endosome fuses with the cell lysosome to create an endolysosome. The whole influenza virus is then degraded by the enzymes contained in this new cell compartment. Thus, the fusion inhibitor compound has played its curative role by preventing viral multiplication in the infected organism.